When the God created us, they gave us a very nice present. They gave us an immune system in order for us to survive. But I also believe that a few months ago, Lord Ganesh called Muhammad Rela and said to him, get this guy from Philadelphia to talk to us about basic of transplant immunology to a bunch of surgeons and hepatologists who are interested or not interested in the immune system. I think that surgeons and hepatologists, medicine people who are dealing with transplant, should be interested in the immune system, should know some basics about the immune system, because knowing these some basics are going to help us to manage these patients. We hear about minimization, we hear about optimization, and so on, but all this should be done once we understand a little bit about the immune system. And what I'm going to do is go over some basics with you. I try to do it in a more simple way, also for me to understand what's going on. I'll focus on the liver. That is the first part. And in the second part, what I'm going to do is try to tell you or try to show you means to measure what I call the temperature of the immune system. And in this way or another, a little bit disagree with the notion of minimization of immunosuppression. What I'm going to try to say is how to measure the immune activity in each one of us, and I will try to show you that. So let's start with kind of like the immune system in the liver. The liver is a very interesting organ because it has a lot of cells that are functioning, hepatocyte, bile ducts, and so on. But it has a lot of other cells that are cells of the immune system. Lots of endothelial cells that can respond, Kupffer cells that are there in the sinusoids that can respond to immune response, hepatic stellate cells, and so on and so forth. It's kind of like so rich in immune cells that by itself, it is immune organ. There's no question about it. And if you look at any kind of like whatever is attacking the liver, coming from the gut, coming from the outside, there must be some kind of like system to filter this foreign that is coming to us. And we were created with two types of system to take care of what is foreign to us. One, we call it the innate immune system, and one we call it the adaptive immune system. The innate immune system is all these Kupffer cells, the monocytes, the natural killer cells that are sitting there. The adaptive immune response is more what we call the T cells. The T cells may be the CD4 T cells, CD8 T cells, the regulatory T cells, and a lot of different kind of like T cells that we are talking about. So what is the difference between these two type of immune responses, the innate and the adaptive immune response? The innate immune response is the first line of defense. Believe it or not, it exists also in plants, in amoeba, in whatever exists and alive under planet Earth. It's the first line of response, and it does not generate any immune memory. That means it responds against everything that is potentially attacking it, without remembering that it will face it next time. It depends on some receptors that are built in our genes, and these receptors are recognizing a lot of structures that are common to many pathogens. So look at it in this way. You have some kind of bugs entering the liver from the gut. These bugs are facing the Kupffer cells here, and these Kupffer cells have to respond. And the way they respond, they phagocyte these bugs. They fight against them by lysing the DNA of these bugs, whatever other proteins and so on. So they activate a lot of things that are innate to them, that are embedded in them against these foreign bugs, also foreign antigens. And at the same time, and we'll talk about it a little bit, this immune system has to be regulated how to stop this attack. The liver immune system is very complex, but it's more interesting for us and more relevant to us when we're talking about, when we're talking about this immune system, is what is more relevant to the transplant setting is the alloimmune system. Before talking about transplantation, there's a very interesting issue to think about the liver specifically when we're talking about the adaptive immunity, something that's associated with alloimmune response. The liver is a very interesting organ. It's not only that it has all these kind of cells, but these cells are kind of like, these cells are constantly under attack from the gut, from a lot of other blood-borne organisms. And let me kind of like describe to you how this system respond when something like a virus get into it. And the easier thing to do with hepatologists is to describe how hepatitis C virus get into the liver and how the liver is trying to get rid of hepatitis C virus. But by the way, hepatitis C virus is easy to explain because you all know about it. It's the same thing if you have alloantigen that is entering into the system. It's not very different. So this hepatitis C virus is going to be broken down to many parts, and some of these parts are going to be presented to T cells, as you see here, directly by a T cell receptor. And there are specialized cells that are going to recognize this complex, the T cell and part of the virus, and once these cells are activated, may it be within the liver or outside the liver in lymph node by dendritic cells, all these kind of now T cells, part of the adaptive immune system, are going to activate response to this virus particle, to this antigen. These cells are going to start to proliferate, and these cells now are going to try to kill the infected cell. These cells are going to try to kill the foreign cell. The foreign cells, in the case of transplant, is a transplanted organ, right? So how this, by the end of the day, is regulated? You cannot attack a system without having the ability to somehow reduce, somehow control, somehow at some stage kill your response against the system, against what is activated. So if we take the innate immune response, the macrophage or so, at the same time that it is chewing the bug, killing the bug, there are a lot of other cytokines that are being secreted, such IL-10, TGF-beta, and so on, that are suppressing its own activity. So that is the innate immune system. In the case of the adaptive immune response, in the case of T cells or so that are activated, if we have cytotoxic T cells, if we have helper T cells, in order to control them, at the same time that these systems are activated, we have activation of regulatory cells, and the regulatory cells are suppressing the activity of this killer T cell, these cytotoxic T cells. So at the same time that there are activation, there is an attempt to deactivate the system. So in the liver, in addition to what I described, the liver is controlling other cells in a very interesting way. It caused a lot of infiltrating T cells to die as they are entering the liver. The exposure of the liver to gut endotoxin and other pathogens all over prepare the liver actually to be more tolerant to foreign, again, all this kind of stimulation lead to local pathological inflammation in case you don't have regulation, and if you don't have regulation, inflammation lead to fibrosis, cirrhosis, and what we know of liver damage. If I had to kind of like ask all of you to really remember one thing about immune system, and one thing about immune system in transplantation is probably this slide, and it's probably the concept of self, non-self recognition. So my immune system is built to recognize only me. My immune system does not recognize Dr. Asher as Dr. Asher, Dr. Ruella as Dr. Ruella, Dr. Patrick Kamas as Dr. Patrick Kamas. It doesn't care about all these individuals as individuals. It cares about me, and it knows only me. So all these individuals, all of you, are simply foreign to me. So once my system sees any one of you, it doesn't care whether you are you as individual, you are you as all foreign to me. And once it sees you foreign to me, it simply rejects you. And this is a very, very important concept in terms of self, non-self recognition. As long as I know myself, I have to remember only myself. All what is put in me, from whoever, it's foreign, period. And once it's kind of like put in me, my system is activated against this foreign antigen, foreign cell, foreign organ, and so on and so forth. Now, here we are. We have a liver. This liver is transplanted. This liver is transplanted, and immediately there are cells that are starting to recognize this liver as a foreign. These cells are maybe dendritic cells, maybe other T cells, and the system is activated in sequence in order to reach to a stage where it is rejected. Now, the basic scientists put this kind of slides. Why these kind of slides are important for us, again, as surgeons and hepatologists? They tell us there are signals, and there are some kind of sequence in which these signals are coming, 1, 2, 3, and 4. And when these signals are activated, by the end of the day, you have rejection. Why it's important for us to understand these signals? Because understanding this kind of pathways, or knowing this kind of pathway, or knowing the concept of this kind of pathway, help us to design immunosuppression. So, for example, if you are taking calcineurin inhibitor, you heard about tacrolimus, you heard about cyclosporine, if you take steroids, we know, based on this kind of pathway, where they are acting. And if we know where they are acting, we know how to use them. We know that tacrolimus and steroids are acting pretty early on in the activation of immune response. We have to give them first and fast in order to inhibit this kind of process. We know, for example, that MMF, you heard about MMF, you heard about Imuran, azoxystrobin, are acting a little bit later on. And actually, if you are going to start to suppress someone with Imuran, you can't expect very, very early response to it. It takes time, and it acts a little bit later. But we, again, know this kind of pathway, and we know how to use this kind of immunosuppression, and we know more than that, how to design new immunosuppression. So, for example, OKT3 and the anti-IL-2 receptor antibodies were designed since we knew that antigens are kind of like attacking here the T-cell receptor. So people design antibody in order to block this very first step. People design antibody in order to block that signal 3, third step. So knowing this kind of pathway is important to know how to design the next drug. And by the way, it's done for any kind of like medication today. Once you know the pathway, you design the drug to attack or block this pathway. Now, what is the difference between us, say, recognize tetanus toxoid antigen versus me recognize Dr. Althoff kidney as a foreign? So I have about 1 out of a million cells that is recognizing tetanus toxoid. But I have 1 out of 20 cells that is recognize a kidney that I'm getting as a foreign body. So obviously, the immune response against alloantigen is much more vigorous than the immune response against any kind of bacteria. And for that, we have to use quite a lot of immunosuppression in order to kill all these kind of cells that are there recognizing alloantigens or recognizing foreign organs. So understanding where and how this immune system is activated and when this activation happens help us to design what to do. We have from the beginning a stimulus, going to activation of T cells, going to division, expansion of these T cells, go to stage of rejection, and we know when this is happening. Times 0, 1 hour after transplantation, 24 hours after transplantation, 3 days we start to see effector T cells, and by 7 days and on, we have the buildup of memory T cells. So we want to start to suppress this kind of response from the beginning, from the first hour, from the first day, and we have to build up our immunosuppression around this kind of pathway of activation. And then we are going from the first stages to maintenance immunosuppression. We heard a little bit about it from Dr. Leroux, and how to prevent the more chronic allograft dysfunction. The problem is that we don't know how to measure this kind of immunosuppression, how much we have to give. We don't have a thermometer to measure this immune response. John, Robert, can you stand up for a second? So look at John and look at me. We are very different. He's a giant person, right? He has hair. He's coming from European ancestry. I'm coming from a different kind of ancestry. So say that you put a kidney or liver in John, and you put the same organ in me. How do I know how much of immunosuppression John needs versus how much of immunosuppression I need to control the rejection against the same kidney? So you can say, maybe he needs more, maybe I need less. How do I measure this kind of immune activation? I think it's very difficult to do that, and right now what we do is really by clinical trial. We give me and John 3 immunosuppression, and then we take one and see how we respond. We minimize the second, and we do and we see how we respond if someone is developing rejection or so. But these are kind of like primitive system, really, to measure the temperature of the immune system. So what I want to kind of go into some more maybe promising, maybe sophisticated way to measure whether John's immune system is more activated than my immune system, or how much is anyone's immune system is activated. And I'm going to show it to you in 2 kind of settings, one in the kidney and one in the liver. Actually, in the kidney, we know much more because the kidney guys were always much better than us learning about the immune response, and maybe we should learn from them. And then I'm going to go to the liver setting. So let's go to the kidney setting. This was an NIH study that I had some time ago. It was multi-center study from 500 patients. So this is industry-style study, a lot of patients. And what we did was to take these patients and just ask them to donate urine at predetermined interval. This kind of arrow here, the white arrows are predetermined interval over the first year after transplantation. And then the patient came to us with rejection. So at the time of rejection, this yellow arrow, we took biopsy. That is standard when you have rejection. And we also took urine sample. And we measured in this urine sample 3 different mRNA levels. Okay, we measure a lot, but we focus eventually on 3 different RNA levels. Now, you see the difference between those who had cellular rejection, who had no rejection, and who had stable rejection. And you think that this difference, when you are measuring mRNA, is almost nothing. That's what we see in this kind of slide, right? But the scale here is very misleading. The difference between here and here is logarithmic scale. So those who had rejection had 10 times more, from 3,000 copy to 30,000 copy of this specific mRNA, which in this case was IP10. We measured CD3, we measured S18 protein, but the important thing that we were able to put together some formula, and again, the difference here is amazing, from 3,000 copy to 30,000 copy. It's not from 3,000 to 3,300. And what we came up with is a sensitivity and specificity curve, what it's called ROC curves, to show you that we have quite sensitive means to measure whether we are dealing with rejection or not at the time that the patient came to determine whether rejection, whether the patient came and had a biopsy. And what we were able to show that we have as a sensitive marker to determine rejection as the biopsy, and maybe even better than the biopsy. So we don't need biopsy anymore to know whether we have rejection or not. Actually, we added the urine metabolite test that made it so sensitive. In fact, we have specificity of 84%, sensitivity of 90% if we combine urine metabolite with mRNA, and it's far, far better than to look at biopsy and to determine whether you have rejection or not. So we did the same with liver patient. Liver patient, we couldn't take urine, we couldn't take bile, so we look at the serum and we look at the serum and measured microRNA. We measured plenty of other things, but I'm going to focus on microRNA because this was the most promising stuff. And similar to what I showed you in the kidney, we had a different predetermined interval during the first year after transplantation, measurement, just random. And at the time the patient had rejection, we had microRNA measurement and we had a biopsy. And similar, we were showing with two different RNA profiles, we were able to diagnose rejection in the liver setting that corresponds to biopsy, and we're able to differentiate this rejection from any other liver inflammation that gave you high AST, ALT. So we have in the liver setting also means to determine rejection. Why it's important as a thermostat for the immune response. Now, these are patients who had rejection versus patients who had no rejection. The rejection happened here. This is the liver, this is the kidney. What we're able to show, not only that we can measure and determine rejection at this time, at the end of these slides in the kidney and the liver, we're able to show that about 20 to 30 days, and this is amazing because it's the same in the kidney and it's the same in the liver, 20 or 30 days before rejection, we can start to see split in the kidney and in the liver, predicting that rejection is going to happen. And it makes sense, because before rejection is going to happen, you're going to have molecular rejection that we don't see yet. So think about what this kind of potential give us, knowing before, knowing when we have molecular rejection. Actually, we can titrate the immunosuppression, reach to a point where we start to see molecular rejection, and at that stage, just stop titrating the immunosuppression. So look at that. These are patients where, kidney patient actually, where they never had any rejection. Patient who had higher creatinine by biopsy did not show rejection, and patient who had rejection. You can see the difference between these two, and this difference is huge in terms of the amount of T-cell mediated mRNA that we can measure in the urine. So we can take these patients, similar to what we can do with the liver patient, and we can start to, how Dr. Olaroth say, to minimize the immunosuppression. And if we minimize it, if we start to go down, we will know where John is going to start to reject the kidney, and maybe it's going to be earlier than me, because once the kidney or once the liver is rejected, this kind of curve starting to behave like this. It's gone up. So we know where to stop this minimization, how to tailor this immunosuppression based on individual number that we get from each patient. So from this time on, we don't have to start to play how much immunosuppression and what kind of immunosuppression combination we need. We have some kind of means to measure the molecular immune response, the activation of the molecular immune response before things can happen, and based on this, to know how much of immunosuppression patient A need, patient B need, and so on and so forth. Maybe one need more, one need less. So I think that genomic markers, metabolomic markers, proteomic markers, they are there. They are there for us to start to experiment and to start to see whether they can be applied in the patient population that we have. In the urine, in the kidney world, I do believe that we have enough of data now to use this kind of markers, to use this kind of genomic markers to determine when there is rejection without biopsy, for sure, no question about that. And we have enough of profiles to determine how to adjust immunosuppression. In the liver setting, I think that we need maybe a little bit more expanded study. This study that we did was on 275 patients. It's a lot of patients, but we need more, and we need from different kind. This study was done in 6 centers. We may need even more centers in order to determine whether microRNA and other metabolites are able to diagnose for us rejection, but what's more important, to measure what I call the temperature of the immune system, the temperature of this alloimmune activation, in order to know what to give to, again, patient A versus patient B. This is Philadelphia. I don't know whether it's looking like Chennai or not, but the last 3 days that I'm here, I'm closed in this hotel. I don't know how Chennai look like. I probably should do it this afternoon. Thank you so much.